Thank you very much for the opportunity to present. It's actually the first time this company is presented, and I'm delighted to do it in the meeting on the MAD, the R meeting. The presentation before just highlighted the message that MSCs have some efficacy, but they need a booster. So the question is exactly the same. How do you improve that efficacy that people have doubled? So the mission is to develop an off-the-shelf, it's a shame we can't really see the presentation there, an off-the-shelf approach with engineered MSCs, with CXCR4 for target and IL-10 for function. We have a team of founders, a team of management and advisors, and we have an initial seed funding, and we're seeking additional seed funding. Very briefly, myself, the CSO, the CFO, and people that will join, making the bridge between the research center, CMAT, in Madrid, so not very far from where we are. Good, now I can see the slides now. And together with several that we're collaborating to develop the product already. We have an international panel. This panel is placed in terms of providing advice on MSCs, engineered MSCs, on GvHD, on IBD, on iPSC-derived MSCs. So clearly a vision to develop an enhanced product with an enhanced functionality, taking advantage of research and expertise globally to deliver that product. We have the assets that are protected by IP on the virus itself. We have the opportunity to use research in lab institutions. We have collaboration with the clinicians to conduct the clinical trial. And the company has been created just about a month ago and will exist between Spain and France. You know about MSCs, you know about IL-10, you know about Tregs and Bregs. But you also know that MSCs take time to deliver that function. And so what we want is to boost that IL-10 production. And coupled with CXCR4 expression on the surface, that transduction will really make the cells go where they need to go and deliver what they need to deliver for immunomodulatory effect. And then we've talked about GvHD before, and why GvHD? The unmet medical need remains. About half of the patients with acute GvHD fail steroids. Half of those patients fail Ruxa. And so there's about 25% of the patients without an opportunity today. We know that GvHD has some opportunity for MSCs. They've delivered some efficacy. They've actually been approved, but they have not given an outstanding therapeutic benefit. We have the opportunity to address those 25% patient need with an increased potency product. There are nevertheless several in development. I just mentioned you the key perspective and why GvHD is a clear opportunity for MSCs. You've seen this before, the data published, the dose-dependent relationship. So clearly, MSCs touch efficacy. Let's bring that power beyond that to deliver an effect. And what is the data that we have with our product to support that? We have a manufacturing process that is pre-GMP. It goes from a collection of adipose tissue and MSC isolation using a established process and the center that is already delivering for a commercially approved product in Europe. Then there is there no, there isn't. Then there is a process of transduction and insertion of gene for CXCR4, IL-10, further expansion, formulation, cryopreservation, and then readiness to use at the level. It is important also to understand the challenges of manufacturing for MSCs. It's not the number, but it is the consistency and above all, the release criteria and the potency. If we have something that we believe is key for the action, I mean IL-10, and we can assay it in vivo and in vitro, we're able to get a prospective potency assay addressing the challenges that we know with the FDA and other regulatory agencies. Now, what is the data that we have supporting the activity and the characterization of this product? You can see there the gene, the CXCR4 IL-10, and this is a proprietary construct by the conjugation of the two, or the combination of the two together. And clearly we've documented on the MSCs the CXCR4 expression on the surface. Okay, check. We have documented the increased IL-10 production, check. And we've documented viral copy numbers within the regulatory expectations. So we do have an opportunity to have an engineered MSC product with a targeted function. Nevertheless, they remain, and we want that, remain MSCs. And the point was also made before, the safety profile of the MSCs through thousands of patients exposed to MSCs, it's important to capture that. MSCs have all that experience. They're a fantastic tool. They're the workhorse of cell therapy that we can now professionalize for a therapeutic effect, remaining the characteristics of the MSCs. Those engineered cells, we have documented in vitro that there is an MSC increased migration on the cells that have CXCR4 on a transwell in vitro assay that you see on the left. And on the right, you can see an increased immunosuppression effect of IL-10. So let's say we check the box of in vitro evidence of a benefit from migration and the benefit of functionality in terms of immunosuppression. If we then go for a non-specific inflammatory process model, we're able to see that only the two that have CXCR4 and IL-10 give the best benefit. Hypothesis, we do have an opportunity to target and immunomodulation. Then we go to a in vivo GvHD model, and clearly we have the untreated animals, and then the MSC naive treated, and a further benefit on top of that for the doubly engineered MSCs. And then we actually take that and do a series of mechanistic studies, and you will see a series of slides here with all the same color code. Black is untreated, gray is treated with unmodified wild-type MSCs, and red is doubly transduced MSCs. And good. And you can see there that if we look at the peripheral blood and the cells, we have a reduction of human cells, but they're all lymphocytes, but they are predominantly T cells with a regulatory function. If we look at the cytokines, again, human T cells, now human cytokines in the blood, we see an anti-inflammatory profile with a reduction of pro-inflammatory and increase of anti-inflammatory cytokines. If we then go and look at the spleen, we see a similar pattern, but now with a B cell component. So if we take all this together, we have good transduction, we have a robust allogeneic donor-based manufacturing process, we have in vivo animal POC, we have mechanism of action data, and we have other elements that enable us to go on to the clinical trial. We also have, and I am not showing here, already animal data on IBD, so already having an opportunity to go beyond with this product, even though there is a perspective of portfolio. So what do we want to do with the company? We want to create it, and it's been done. We want to complete the R&D and get ready for the GMP stage. We want to go into the clinic within the next 18 months, and we're already actively working on that. But we want to use this as a platform to use MSCs to be engineered. And you've seen that before, and you're going to see more and more of that. That there is a new era that we are generating for MSCs, using them with the tools that we have today to improve their functionality. We have a detailed plan that includes R&D activities, GMP process. regulatory interactions, clinical stage for the activities that we want to obviously implement for the clinical study. And we have a strong collaboration with the University of Navarra with a intention of a clinical study already with an institutional grant to execute it. So clearly everything in place to be in the clinic on the timelines that you can see there. In other words, patients included in the study on the second half of next year. That study will be on acute GvHD for patients non-eligible or failing Ruxo, where the unmet medical need is, where the opportunity is. You can see the outline there. Obviously, we're perfecting this, and we will finalize it upon regulatory discussions. But we have a vision to go beyond that in terms of using the MSCs, also perfecting the source, i.e. considering iPSC, but including additional genes for additional functions. And so clearly a layer of immediate product, multiple indications, but a platform of genetically engineering the MSCs for a purpose. So we have a technology platform close to the clinic, a corporate operating ability, and we're seeking funding on top of the seed funding that we received from HadBio partners, who actually has been instrumental to put the company in place and brought me on to execute that. And so we need to go beyond that. That's the perspective of where we want to go, the seed extension, be ready for a Series A, where we aim to be, once we have the clinical data from the current study, and the perspective of the platform. So very simple. MSCs do have some efficacy. MSCs can be improved by genetically engineering, targeting, and function. We have all the preclinical and the operational capabilities, seeking additional funds to be able to go into the clinic. The deliverables will be to have an industrial-grade product by the beginning of next year, be in the clinic the second half of next year, have data on the course of the following year. Takeaway message. I mean, it's a simple message. MSCs have great cells. MSCs are amateurs. We need to professionalize them with the technologies we have today. And we're doing this between, and I wasn't sure if I was going to have time, but I will labor on it. We are doing this between France and Spain. I don't know if you know, but there's an island between the two countries that is 6 months French, 6 months Spanish. I mean, it's Europe, right? And it's called Pheasant Island. But actually, a nicer way to say it is Kiji, which is in Japanese for pheasant. So that's why the company is called. It's also the bird and the rebirth of MSCs as we see it. I'll stop there, and if Elizabeth wants, I have time for questions. Is there any question? You can always talk to me afterwards. If I survive the hospital exemption panel this afternoon. All right, thank you.